So in this video, we're going to talk about the CRISPR-Cas9 system. CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats, and they're the defense system of prokaryotes, almost like their immune system. And what this CRISPR looks like in the DNA is that you have repeats. They're 20 to 50 base pair longs. And in between these repeats, you have sequences of DNA called Proto Spacers. And these Proto Spacers are DNA segments that are identical to bacteriophage DNA, or viruses that attack bacteria. And these are records of foreign DNA that this bacteria has encountered, or the lineage of this bacteria has encountered and survived through. And they're a way to recognize if that bacteria gets attacked by the same virus, it can destroy that viral DNA. And any DNA that's at the 5' end of the CRISPR locus that's a protospacer would be the latest virus that bacteria has encountered. Now, the way that CRISPR works is that this locus is transcribed to make a single long RNA. Then this RNA is transcribed and then processed to form roughly 30 nucleotide transcripts that are known as cRNAs. So you can see the cRNA is made out of the Proto Spacer and the CRISPR repeat that has been cut. Now, these cRNAs use a protein called Cas to find their complementary DNA and cut that complementary DNA by invading the double-stranded DNA and inactivating them. And this is, since this is the immune system for prokaryotes, the bacteriophages are mutating to avoid being targeted by these cRNAs. Now we're going to look at the type 2 system of CRISPRs, which only need the protein Cas9 to work. So Cas9 has nuclease activities, so they have HNH-like domain that cleaves target DNA that's complementary to the cRNA. And it also has a RucV-like domain that will cleave DNA on the opposite strand as well. So we can see that in this image, HNH will cleave on the target strand, RucV, it should be RucV, not RucV, targets the non-target strand. So for this Cas9 and CRISPR system to work, you also need another piece of RNA called the tracer RNA, or trans-activating CRISPR RNA. Now, tracer RNA will bind Cas9 and will form a base pair with the cRNA. So that's what's being shown here. So our tracer is binding to the Proto Spacer, which makes sense because the Proto Spacer is the same no matter what cRNA you are using. And this helps to interact with Cas9. So once you have the tracer and the cRNA bound, then Cas9 will go and it will find the target DNA if this target DNA has what's called a PAM, or Proto Spacer Adjacent Motif, on the 3' side that's to be cut. And so the reason why we're looking for another DNA sequence other than what is complementary to us is because if we only cared about finding DNA that's complementary, we would go back to our own DNA where our CRISPR repeats are, where our protospacers are, and cut our own DNA. So by looking for this PAM, it's a safety mechanism to make sure we only cut viral DNA. And the PAM is generic. So for example, in the bacteria Streptococcus pyogenes, the PAM is just any nucleotide GG. So as long as you have a GG with something in front of it, that's what we're looking for. And this happens every 8 base pairs on average. So eventually, not too far away from our 3' end will be this PAM sequence, and then we can cut it. And remember, the HNH will cut the target, and the RUV-C will cut the non-target. And people have used this CRISPR-Cas9 for gene editing. And we do that by creating what are called sgRNAs, or single guide RNAs. And what a single guide RNA is that you take your CRRNA and you now covalently link it to your tracer RNA. So it's one RNA molecule now. Previously, they were just hydrogen bonding here, but instead of doing that, now we just connect it through with a linker so they can never be attached again. And so when this guide RNA with this Cas9 come in, they will find the target they're looking for and they will cleave. Once cleaved, your cells will try and repair this double-strand break via 2 methods. One, they'll do non-homologous end joining, which will mean that this part that was cleaved will try to be fixed, but it's going to create insertions and deletions, which means if you cleave a gene, you're not going to be able to fix it correctly, and you will inactivate that gene. Or they're going to do a homology-directed repair, where let's say that you target a gene and you also supply DNA that's homologous to that region. So what will happen is that the cell will try to do a homologous directed repair, and you can insert a new gene or a mutant version of that gene right into the genome, and you have modified the organism's DNA. Now you can be even fancier with this mechanism, and you can use 2 different guide RNAs that can target 2 different parts of the cell, and you're making double-strand cuts, so you're going to cut off a large portion of the DNA if you want. So this is a way to delete or replace DNA segments between sites. What you can also do, instead of just cutting the DNA, you can attach an effector to Cas9, and now Cas9 no longer cuts, but it will just sit on that gene, which will inactivate that gene because there's a large protein sitting on that DNA. That DNA can no longer be transcribed, so it can be a repressor of that gene. Or what you can do is you can make this guide RNA be further downstream of where the gene is, attach it to an activator, and now you're going to turn that gene on because this activator will make sure this gene is transcribed at a high rate. So that just shows you the utility of this CRISPR-Cas9 sequence. And one other thing you can do is you can attach GFP to Cas9. So let's say you know there's a particular gene you want to look at, you're not quite sure where it is, you take your guide RNA to target that, you have your Cas9 modified so it doesn't cut, you stick green fluorescent protein on your Cas9, then you send it into the cell, and green fluorescent protein or GFP will fluoresce underneath a microscope, and so you can target where in the cell, where in the genome is that gene. So that's just all the utility of the CRISPR-Cas9 system. And that's it for this video. I will see you in the next video.